Okay, so this is my final workshop. It's all about DNA and how interesting DNA can be in everyday practices and everything. And there was going to be a guest speaker, but she was unavailable for today because she has a presentation elsewhere in Ohio. So she couldn't be available. She has a presentation at the exact same time as this. So she couldn't be here, but I do have some information about her, and I can put you in contact with her if you would like later on. She's a doctor. So first, let me talk about what DNA is, then the contributors to DNA, specifically Rosalind Franklin, DNA fingerprinting and cloning, then cell cycle regulation, the errors in cell duplication, and just the applications of genetics. And this is no longer with the person. I forgot to change the name over there. But so DNA. DNA is an abbreviation for deoxyribonucleic acid, and it contains a pentose sugar, a phosphate, and a nitrogen-containing base. So where the sugar is in the middle and the base is on the sides and the phosphates are on the outside. So the base pairs are connected to each other, and so the nitrogen-containing base can either be a pyrimidine or a purine. So the pyrimidine would be a 6-member single ring, so it's only the 6 carbons, and it can either be thymine or cytosine. And in RNA, instead of thymine, there's uracil. And in the purines, which is the 2 rings, so it's 1 6-membered and 1 5-membered ring. And those ones are connected to the pyrimidines, and they are adenine and guanine. So in a cell, DNA can help with replication, so it can help pass on different traits to different people, and it's basically combined through the genes of the parents, so the female and the male that are creating the person. And DNA also helps control cell activity, so the growth and metabolism of the cell, so it can help regulate with the cell checkpoints. And then there's mutations that allow for permanent changes in an organism to be passed down. So DNA can be mutated to change its sequence, either for better or for worse. Mutations can be good and bad. Certain mutations are beneficial, or they just change the person. Some of them have no effect. Some can be beneficial. Some are detrimental to humans. Like a mutation that has no effect would be like blue eyes. That has no effect on humans. It doesn't change anything, but it's just a mutation that humans ended up developing. So some significant contributors to DNA are Rosalind Franklin, and also Alfred Hershey and Martha Chase basically created the foundation for a lot of DNA research. So they proved that DNA is genetic material, so they used E. coli cells or bacteriophages and essentially labeled the stuff inside. So it's labeled the DNA, and they labeled the capsule or the protein, and then essentially they allowed the bacteria to get invaded by this the E. coli to get infected by the bacteriophage, and then they saw what was being released into the actual cell, because that would be what contains the genetic component. And they saw that once it was infected, that the DNA was what was being injected into the cell, meaning that that's what contained the genetic component of DNA, of like creations and of proteins. Then there was James Watson and Francis Crick, and they showed that DNA is a double helix, and it's formed in a double helix, and they showed how they're meant to be connected. So they conclude that the sides are the sugar phosphate backbone, so the phosphate's on the outside, while the nitrogenous base pairs are in the middle. And they also concluded that the bases are adenine, thymine, guanine, and cytosine, so where adenine and thymine are together and cytosine and guanine are together, meaning that these strands are anti-parallel, and that the pyrimidine and purines connect to each other, and it's not anything else. But someone who's quite unsung in the DNA research is Rosalind Franklin, who provided much of the research that was instrumental to Watson and Crick's discoveries. And she helped produce images of DNA that were never seen before, like Photo 51, which got very famous because of this. She also helped conclude that DNA was anti-parallel and determined that the logical base pairing of it. So a lot of the research that previously was done was actually developed through Rosalind Franklin's research. So she concluded that purine and pyrimidines are connected together because of the width of Photo 51. So she was able to measure and see how the different widths, the widths that are present in a lot of DNAs are only possible if purines and promines were connected together and nothing else. So she helped differentiate between A and B forms of DNA, which helped further other research teams' projects. And the majority of her research was overlooked and taken from her by Watson and Crick, which were competing scientists that wanted to basically achieve the research faster than her. So they took a lot of her research and then published it under their own names. Then DNA fingerprinting and cloning. So DNA fingerprinting is essentially using sequences of DNA to identify people or identify things that have DNA. So it also allowed for identification of people and criminals through DNA strands. It was developed by the British geneticist Alec Jeffreys. And so the process is to take a DNA sample, so hair, skin, anything that contains part of the genetic material, including blood, and to treat the DNA with a restriction enzyme, which cuts the strands at specific nucleotide sequences. So since everyone has different sequences, the restriction enzyme will cut at different parts, meaning that every single person has a different kind of layout of how it's going to be cut. And each DNA strand has an overall negative charge. So these fragments are inserted into a positively charged agarose gel. And this gel is where gel electrophoresis takes place. So it moves the negative charged DNA down because of its charge. And when DNA is larger, so if the sequence is a longer sequence, it'll get caught on the clumps more. So the smaller DNA will move further down and will get stuck near the bottom. So it creates different levels in the strand placement. So like as you can see, every single person has a different number of fingerprints and different number of places where the DNA stops. So then when you have a suspect and you have an actual blood stain, you can actually see who is exactly like that blood stain. So that would be the person whose blood you have. And also there is cloning, which is a process where you can produce identical copies of molecules, genes, or even entire organisms, like they cloned the sheep Dolly. So what you would do is use an unfertilized egg and enucleate it through inserting a tube into the cytoplasm. So removing the nucleus from that unfertilized egg. And this enucleated egg is fused with a nucleus from a cell that you want to clone. So whatever you want to clone, you get that nucleus and you insert it into an enucleated egg. And this cell divides normally, continues this process, and then is injected into a surrogate. So any other animal that can carry this egg will take it, and so the surrogate will carry a clone of the nucleus that was injected, nothing else. So it will result in an identical clone of whatever provided the nucleus. And as you can see, it was shown it was done first in a sheep, and they actually created a successful clone. But many clones don't live until the same age that the actual original animal would. They usually die pretty early on because of just how cells work. And even though you can create a perfect clone of it, it usually never ends up being as successful as just a regular animal would be. So then there's cell cycle regulation. And so the cell cycle has cell checkpoints, which generally go through every single cell, goes through all these checkpoints all the time. So any eukaryotic cells, so the cells with nucleuses, are controlled by the molecular control system. So there's a couple checkpoints. So G1 is the first checkpoint at the end of duplication during interphase. And this checkpoint checks for cell size, nutrient amount, growth factors, and DNA damage. So if any of that happens, so if there's DNA damage, for example, they will be taken out of circulation and will basically be able to restart as a new cell, but not continue on. And if the cell is at proper size, it'll continue. And if it has proper nutrients and growth factors, it'll continue. This is also the restriction point. So if there is any DNA damage, the cells can fully exit the cycle without being destroyed. And then in the G2 phase, after DNA duplication, so checks if DNA is duplicated properly. And then after this checkpoint, the cell becomes committed to mitosis. And if there are any defects, the cell can pause and fix the problem or go through apoptosis, which is essentially just programmed cell death. So once a cell has a problem, it no longer can exit. It will be destroyed. And then in the M cycle checkpoint, this is the spindle checkpoint after anaphase, so after the spindle fibers are attaching or meant to be attached. And if the microtubules are not attached in proper places, the cell will pause to allow for attachment, and if they are, it'll continue. And if there's no way of connecting the microtubules, then the cell would once again go through apoptosis. The cell cycle also works on cell cycle clocks, which are make it much more complicated. So there are cyclins and cyclin-dependent kinases, which are one of the combinations for maturation promotion factors. So cyclins are proteins that drive the cell cycle forward. And there are 4 types of cyclins. So G1 cyclins, G2 are S cyclins, and M cyclins. So they all are for the specific part in the checkpoints. And so cyclins are cyclical, so they rise and fall. They're not constant in amount. And they help phosphorylate other proteins. So they basically allow phosphor groups to attach to the proteins that they want to activate. And so cyclin-dependent kinases are proteins that get phosphorylated by the cyclins, and they are in constant amounts, but they're only useful if the cyclin is high enough to actually phosphorylate them. So it's kind of a handover thing where the cyclins have to give off the phosphorus so that the cyclin-dependent kinases can actually use it. And so maturation promotion factors are a combination of CDK and M cyclin, so a specific type of cyclin, which is the M cyclin. And this is during prophase, and it helps open the nuclear envelope and promotes the condensation of chromatin into chromosomes, allowing mitosis to happen. So that's why it's called maturation promotion factor for M cyclin. So it's mitosis cyclin, kind of. And you can also see just how for different types of cyclin, there's different lengths of phases and different concentrations. And each one has a different amount. So there's also the errors that can happen in cell duplication. So mitosis and meiosis both can function properly, or they can have a problem that would usually cause apoptosis. But if it's not seen, it can cause abnormal cells and cause excessive growths that turn into tumors. And even regular cells can face these issues while duplicating. So during anaphase 1 and 2, so anaphase 1 is during mitosis, and anaphase 2 is only relevant in meiosis, but meiosis has both. It's possible that the microtubules don't properly connect to the parts of the cell, so it won't separate into chromosomes. So when the cell divides, one side has monosomy, another one has trisomy. So there's one chromosome and three chromosomes. And if this happens, then the chromosome number will be off in the future offspring of that cell, meaning that it can cause different issues in humans, especially. So, for example, the 23rd chromosome determines sex in humans, and if there's an extra, so if there's trisomy, it'll turn into Klinefelter syndrome, which affects males, and if there's only one, so it's monosomy, it'll become Turner syndrome, which affects females, and both of them end up with they cannot reproduce, and they end up with different traits that could be similar to the opposite sex. So either the male with Klinefelter syndrome would be more genetically similar to a female, while the female with Turner syndrome could be more genetically similar to a male. And also there are genetic diseases that can be passed on through people and through DNA itself. So certain diseases are passed on from parents to children. So some of them are autosomal recessive, meaning that both parents have to carry the trait so that the disease actually gets passed on. And if it's recessive, that means that it's not common, or it will not happen if both parents don't have that trait. So some examples are alkaptonuria, albinism, and Tay-Sachs disease, all of which can only happen if both parents have a trait of that disease. Another one's autosomal dominant. So autosomal dominant diseases don't matter whether or not both parents have it. Only one parent has to carry that trait because it's dominant, meaning that it will take over no matter what. So some of these are progeria, Huntington's disease, and human achondroplasia. And for progeria, although the people who have progeria cannot reproduce because they end up passing away before they can reach the age of reproduction, it is predicted that these people, if they were able to, it would be an autosomal dominant disease. Although all the known cases of progeria happen because of a genetic mutation, not because of the inherited disease. And Huntington's is only clear, like, people only know what's happening when it's really because of very late onset disease, meaning that many people have children before actually knowing that they have this disease. And it can cause many problems when having children, because the onset's usually after 35. So once people have children, they know they have the disease, and then they have 100% passed it on to their children. And then X-linked diseases are sex-linked, so that means that they affect one sex more than the other. Usually this affects males more often because males have only one X chromosome instead of two X. So the X-linked diseases are X-linked, so they affect the X chromosome side more. So that means that males have a higher chance of it because they either have it or they don't have it, but females can carry it, men cannot be carriers. So some of these examples are hemophilia, color blindness, and Duchenne muscular dystrophy. Two of which hemophilia and color blindness can be like not seen. Sometimes people never know they have hemophilia, and it's been basically eradicated in today's world. And color blindness isn't very it's not very detrimental to a person, but Duchenne muscular dystrophy can be very detrimental to people. So it's much worse when boys are affected with it. And it can cause the lack of growth in a person's muscles, and then cause many issues later on. And then it can essentially kill a child before he is of age to really be an adult. So as technology progresses, there's more ways of sequencing DNA and figuring things out with DNA. So DNA technology helps with the production of vaccines and protein therapies because they can sequence DNA and see what parts of the DNA are relevant in these diseases. So with insulin, DNA can really help figure out how it works and how it's relevant to certain people. And human growth hormone deals with obviously the growth of the hormones and the growth of cells. So once again, DNA research can help see how these hormones affect people who are taking it. And then recombinant DNA technology helps in isolating DNA strands that allows for characteristics to be singled out. So such as CRISPR, it can edit genes and remove certain genetic diseases, which can be very beneficial or very detrimental depending on who has access to this technology, which is why CRISPR isn't very widespread as is. Since this technology can help take out certain genes for genetic diseases, which can be very beneficial if it's something very harmful, but it can also cause a door opening to essentially genetic editing so that children are how people want them to be. So it can be like creating designer babies and things that aren't as centered in helping people, more centered in eugenics. So this is the person I invited today, Nusha Navidez Sunny. She's a doctor and she is currently in Ohio, but she specializes in addiction and also different aspects of medicine. She used to do, I think, more family medicine, then she switched to addiction. And she was going to talk about how DNA affects her personal life, but because she's not here, instead we have a Kahoot, which we're going to run instead. So I can open the Kahoot and everyone can join. I can remember how to do Kahoots. Sorry, I'm trying to figure out how to do this. I'm pretty sure there's not many people here, so I think it'll work because Kahoot has changed a lot. So once it creates the pin, there's only 3 people here, and I think it only allows 3 people, so that's good. So once everyone joins, then we can start. It's just about everything we've learned so far. All right, and then once Zara joins, then we can start it. There we go. So what are the components of DNA? All right. So the DNA components are pentose sugar, a phosphate group, and a nitrogen-containing base. So the other answers had glucose and phosphate group and phosphorus group. And it is the definitions are very important when it comes to DNA. So it has to be pentose sugar, not a glucose, when it's used in DNA. So the next question. So this is true or false. So there are 2 types of nitrogen-containing bases. So this is true. There are only two types, so purines and pyrimidines. All right. So this is just to complete the answer. So purines have a two-membered ring, a single-membered ring, include thymine and cytosine, or include adenine and guanine. So purines have a two-membered ring and include adenine and guanine, so both answers are correct. So yeah, adenine and guanine are wider than pyrimidines are. All right. So this is the same thing, pyrimidines, just finish the question, the answer. So single-membered ring, two-membered ring, adenine, guanine, cytosine, and thymine. Same answers. So pyrimidines include cytosine and thymine and have a single-membered ring. So they're much thinner than purines are. And I think everyone got that right. So what does DNA do in the cell? So DNA controls cell activities, as was shown like in the DNA cycles and everything. So other things control everything else that's up there, right? So who proved that DNA was genetic material? So Alfred Hershey and Martha Chase proved that DNA was genetic material and not that it was protein. So which one is a base pair? So adenine and thymine is a base pair. Everything else is either the same thing or it's not a base pair. This is a true or false question. So Watson and Crick differentiated between the A and B forms of DNA. Is it true or false? So that is false. That was Rosalind Franklin. Who developed DNA fingerprinting? So Alec Jeffreys was the person who developed DNA fingerprinting, and he was British. Which is the clone? Which is the clone identical copy of? So which of the three? Yep. So the nucleus is what the clone is a copy of. Since the nucleus is what provides all the genetic material, everything else is just carrying the nucleus. So what is G1? So G1 is the restriction point, so where the cell can exit the cycle if it needs to, and if it's not perfect. So cyclin-dependent kinases are the rise and fall in a cycle. Is that true or false? So that is false. Those are cyclins. Cyclin-dependent kinases are dependent on the cyclins, but they're constant throughout the cell cycle, throughout the cycle. An extra 23rd chromosome is called. So that is trisomy, means extra 23rd chromosome. Everyone got that right? So autosomal dominant diseases affect one sex more than the other, true or false? These are autosomal dominant. So it is false. Those are X-linked diseases. So autosomal dominant are dependent on the actual DNA sequence, so who carries and who doesn't carry. And last question, what does CRISPR do? So CRISPR helps edit genes to remove diseases. So it is a DNA sequencing tool. So that was it. And just the ranking if everyone wants to see it. So Zara answered the most correct. Good job. And this was going to be a lot longer with the Q&A, but because of the doctor that is not here, because of her presentation, she couldn't make it, so now it's a lot shorter. And this is the end of the presentation.
Does anyone have any questions for Shiley about this? Zara,
Yeah.
bio is my worst STEM subject, so that is funny.
Yeah.
Yeah, bio is a whole other beast. That's why we all respect doctors and anyone in the bio field, because they're a whole other breed, those guys.
I can't do physics, so it's
And that's why you guys respect the
yeah.
rest of us.
Yeah.